Section 1. The building blocks, amino acids. Proteins are composed of 20 standard amino acids, each featuring a central carbon atom, also known as the alpha carbon, an amino group, known as NH2, a carboxyl group, COH, and a distinctive side chain, R group. The diversity in side chains gives each amino acid unique properties, influencing the overall structure and function of proteins. Amino acid structure and classification. 1. Amino acid structure. Alpha carbon, central to the amino acid structure. This carbon atom is attached to 4 different groups, making amino acids chiral, except glycine. Amino group minus NH2, typically protonated under physiological pH, forming the NH3 spice. Carboxyl group, nice COOH, usually deprotonated, giving COO- at physiological pH. R group, side chain, differentiates each amino acid and dictates its chemical behavior. 2. Amino acid classification. Essential versus non-essential. Essential amino acids must be obtained through diet, e.g. leucine, lysine. Non-essential amino acids can be synthesized by the body, e.g. alanine, aspartic acid. Polar versus nonpolar. Nonpolar amino acids, hydrophobic side chains, e.g. valine, leucine, isoleucine. Polar uncharged amino acids, hydrophilic but uncharged side chains, e.g. serine, threonine. Polar charged amino acids, acidic, e.g. glutamic acid, aspartic acid, or basic, e.g. lysine, arginine. Acidic versus basic. Acidic amino acids, negatively charged at physiological pH, e.g. glutamic acid, aspartic acid. Basic amino acids, positively charged at physiological pH, e.g. lysine, arginine, histidine. Peptide bonds in protein formation. Amino acids link through peptide bonds, forming long chains called polypeptides. A peptide bond is an amide bond formed between the carboxyl group of one amino acid and the amino group of another through a dehydration reaction. Peptide bond formation. 1. Dehydration reaction. Removal of a water molecule links the COOH and NH2 groups. 2. Planar nature. The peptide bond has partial double bond character, restricting rotation and leading to a planar conformation. 3. Polarity. Due to the presence of a free amino group at one end, N-terminus, and a free carboxyl group at the other, C-terminus, peptides have polarity. Significance in protein structure. 1. Primary structure. The sequence of amino acids in a polypeptide chain forms the primary structure, which determines how the protein will fold. 2. Folding potential. The unique properties of each amino acid influence folding patterns, ultimately determining secondary, tertiary, and quaternary structures. 3. Chemical reactivity. Certain amino acids contain reactive groups in their side chains, contributing to enzyme catalysis, binding specificity, and structural stability. Examples of unique amino acids. 1. Proline contains a cyclic structure, introducing a kink in the polypeptide chain and reducing flexibility. 2. Cysteine can form disulfide bridges with another cysteine, stabilizing protein structure. 3. Glycine lacks a chiral center and has high flexibility due to its small side chain, H. Functionality and evolutionary perspective. Functional diversity. The chemical diversity of amino acids allows proteins to perform various functions such as enzymatic activity, structural support, signal transduction, and transport. Evolutionary conservation. Key amino acids involved in critical functions, e.g. active sites of enzymes, are often conserved across species due to evolutionary pressures. Conclusion. Understanding amino acids and their interactions is foundational for deciphering protein structure and function. The unique combinations and sequences of amino acids give rise to the vast diversity of proteins, enabling them to act as enzymes, structural components, and signaling molecules, thereby driving all biological processes. References. 1. Eisenhaber-Frank, Prediction of Protein Function, 2 Basic Concepts, and 1 Practical Recipe. Landis Bioscience and Springer Science Business Media, 2006. 2. Additional sources. Section 2. Protein Primary Structure, Sequence Matters. The primary structure of a protein is the linear sequence of amino acids as encoded by the genetic material of an organism. This linear chain, often termed a polypeptide, ultimately determines how the protein will fold into a three-dimensional structure and thus dictates its biological function. The specific sequence of amino acids in each protein provides the basis for its unique identity and function. Genetic code and the central dogma. The primary structure is derived directly from an organism's genetic code, following the principles outlined in the central dogma of molecular biology, DNA, RNA, protein. 1. Transcription, DNA to RNA. DNA, composed of 4 nucleotides, adenine, thymine, cytosine, and guanine, stores genetic information. In transcription, an RNA polymerase enzyme reads a DNA strand and synthesizes a complementary RNA strand, mRNA. The mRNA sequence mirrors the DNA template, replacing thymine, T, with uracil, U. 2. Translation, RNA to protein. mRNA is read in sets of 3 nucleotides known as codons. Each codon specifies a particular amino acid or signals translation termination. Transfer RNA, tRNA molecules, each carrying a specific amino acid, recognize the codon through their complementary anticodon. Ribosomes facilitate peptide bond formation between adjacent amino acids, resulting in a growing polypeptide chain. 3. The genetic code. The genetic code is universal, degenerate, and non-overlapping. Degenerate, multiple codons can code for the same amino acid, e.g. 6 codons for leucine. Start codon, methionine initiates translation. Stop codons, UA, UAG, and UGA signal the end of translation. Sequence analysis, motifs, patterns. The amino acid sequence provides vital clues to a protein's structure and function, particularly through specific motifs and patterns. 1. Motifs and patterns. Motifs. Short, conserved amino acid sequences associated with specific biological functions, e.g. zinc finger, helix turn helix. Patterns. Combinations of amino acid residues involved in catalytic or binding functions, e.g. ATP binding, glycosylation sites. Zinc finger, a cysteine and histidine-rich motif that binds zinc ions, stabilizing DNA binding domains. Helix turn helix, a motif involved in DNA recognition and transcription factors. EF hand, a calcium-binding motif present in many signaling proteins. Walker A and B motifs, ATP or GTP binding motifs in nucleotide-binding proteins. Patterns, N-glycosylation site, recognized by the sequence S.N.X or Thr. Phosphorylation sites, often serine, threonine, or tyrosine residues surrounded by specific patterns. 2. Sequence alignment. Pairwise alignment, comparing 2 sequences to find regions of similarity, indicating evolutionary relationships or functional similarities. Multiple sequence alignment, MSA, aligning several sequences simultaneously to identify conserved regions and infer functional motifs. Scoring and metrics, Blossom 62 and PAM matrices, provide scoring systems for substitutions. E-value, expect value, indicates the statistical significance of a match. Identity versus similarity. Identity refers to exact matches, while similarity includes conserved substitutions. Functional implications of primary structure. 1. Homology and function prediction, orthologs, genes in different species with a common ancestral gene, often retaining similar functions. Paralogs, genes within the same species arising from duplication, potentially with divergent functions. Annotation transfer. Functional information from experimentally characterized proteins can be inferred for uncharacterized proteins based on sequence similarity. 2. Protein families and domains. Domains, independent folding units often associated with specific functions. Families, grouping of proteins sharing one or more common domains. Databases. FAM, SMART, ProCite provide curated domain and motif information. 3. Sequence variability and evolution. Conserved regions indicate critical functional or structural roles. Variable regions allow adaptation to new functions or interactions. Evolutionary pressure. Positive selection for beneficial mutations versus purifying selection against harmful changes. Disease and mutations. Impact on primary structure. 1. Single nucleotide polymorphisms, SNPs. Common genetic variations affecting protein sequence, e.g. sickle cell anemia due to glue, val substitution in hemoglobin. 2. Insertions and deletions, indels. Frame-shift mutations leading to entirely different amino acid sequences. 3. Disease-related mutations. Cystic fibrosis, deletion of phenylalanine at position 5 and 8 in the CFTR protein. Tay-Sachs disease, mutations in the hexa gene affecting beta-hexosaminidase A. Conclusion. The primary structure of proteins forms the foundation upon which higher-order structures and functions are built. Understanding this structure is essential for deciphering the functional aspects of proteins, predicting their roles, and identifying the implications of mutations. Comprehensive sequence analysis is a powerful tool for understanding biological processes and the molecular basis of diseases. References: 1. Eisenhaber-Frank, Prediction of Protein Function, 2 Basic Concepts and 1 Practical Recipe, Landis Bioscience and Springer Science Business Media, 2006. Additional sources: Section 3, Protein Secondary Structure, Local Folding Patterns. Introduction to Protein Secondary Structure. The secondary structure of proteins refers to the local folding patterns of the polypeptide backbone within a protein. The 2 primary types of secondary structures are alpha-helices and beta sheets, both stabilized by hydrogen bonds. Additionally, turns and loops serve as connecting elements between these regular structures, providing flexibility and enabling the formation of complex tertiary structures. Alpha-helices. The alpha helix is a right-handed coiled structure with 3.6 amino acids per turn. The carbonyl oxygen of each amino acid forms a hydrogen bond with the amide hydrogen of the amino acid 4 residues ahead in the sequence. 1. Structure of alpha helices. Hydrogen bonding pattern. Hydrogen bonds form between the carbonyl oxygen of the nth residue and the amide hydrogen of the n+4th residue. R group orientation. Side chains point outward from the helical backbone, reducing steric hindrance. Dipole moment. Due to the cumulative effect of all the hydrogen bonds, alpha helices have a significant dipole moment, with the N-terminus being positive and the C-terminus negative. 2. Characteristics of alpha helices. Pitch. The rise per complete turn is 5.4 awitz. Handedness. Right-handed helices are most common due to lower steric hindrance. Hydrophobicity. The helix surface can be hydrophobic or hydrophilic, depending on the side chain properties. 3. Distribution and functionality. Globular proteins. Alpha helices are prevalent in the core or surface of globular proteins, contributing to stability and functional interfaces. Membrane proteins. They often traverse membranes as transmembrane helices. DNA-binding proteins. Many DNA-binding motifs, such as helix, turn helix, and zinc fingers, contain alpha helices for sequence-specific interactions. 4. Helix stabilizing factors. Amino acid propensity. Alanine and leucine favor helix formation, while proline and glycine often disrupt helices. Helix capping. Certain residues, e.g. asparagine glutamine at the helix termini, can form hydrogen bonds, capping the structure. Beta sheets. Beta sheets are stabilized by hydrogen bonds between backbone carbonyl and amide groups of adjacent beta strands. They can be either parallel or antiparallel. 1. Structure of beta sheets. Beta strands. Individual polypeptide chains with a fully extended conformation. Hydrogen bonding pattern. Hydrogen bonds form between carbonyl oxygens and amide hydrogens of adjacent strands. Parallel versus antiparallel. Parallel beta sheets. Strands run in the same direction, with hydrogen bonds forming at an angle. Antiparallel beta sheets. Strands run in opposite directions, with hydrogen bonds forming in a linear fashion, providing greater stability. Characteristics of beta sheets. Twist. Beta sheets exhibit a natural twist due to the chiral nature of amino acids. R group orientation. Side chains alternate above and below the sheet plane, giving beta sheets a pleated appearance. 3. Distribution and functionality. Globular proteins. Beta sheets often form the core of globular proteins. Fibrous proteins. Silk fibroin and amyloid fibrils consist primarily of beta sheets. Beta barrels. Beta sheets can form cylindrical structures, e.g. porins in outer membranes. 4. Sheet stabilizing factors. Amino acid propensity. Valine, isoleucine, and tyrosine favor beta sheet formation. Beta turns. Short loops that connect antiparallel strands. Turns and loops. Turns and loops are irregular secondary structures that connect alpha helices and beta sheets. They are crucial for the protein's overall fold and function. 1. Beta turns. Type 1 and Type 2, the 2 most common turn types, differing in the orientation of the carbonyl group. Residue arrangement. Turns involve 4 residues, with hydrogen bonds forming between the carbonyl oxygen of the first residue and the amide hydrogen of the fourth. 2. Hairpin turns. Antiparallel connection. Hairpin turns connect antiparallel beta strands. Common residues. Glycine and proline are frequently found due to their conformational flexibility. Loops. Longer connecting elements. Loops range from 6 to over 20 residues and connect secondary structure elements. Surface exposure. Often found on the protein surface, loops are involved in ligand binding, protein-protein interactions, and conformational changes. Prediction and analysis of secondary structure. 1. Prediction tools. Cho-Fasman method. Based on amino acid propensities for helices or sheets. Garnier-Oskauthorpe-Robson GOR method utilizes statistical information from known structures. Neural network approaches. Modern tools like pCypred and JPred use machine learning to improve accuracy. 2. Secondary structure databases. DSSP, Dictionary of Secondary Structure of Proteins, classifies secondary structures from protein crystal structures. SCOP and CATH organize proteins based on their secondary structure arrangements. 3. Functional correlation. Motifs. Secondary structure form motifs like EF hands, leucine zippers, and coiled coils. Domains. Common secondary structure combinations form functional domains. Significance in protein function and disease. 1. Structural stability. Proper folding into alpha-helices and beta sheets is crucial for protein stability and function. Misfolding leads to aggregation, as seen in amyloid diseases, e.g., Alzheimer's. 2. Protein engineering. Understanding secondary structures enables the rational design of proteins for specific functions. 3. Disease-related misfolding. Mutations affecting secondary structure regions can lead to diseases. Sickle cell anemia. Mutation in hemoglobin's beta chain affects secondary and tertiary structures. Huntington's disease. Expansion of polyglutamine repeats forms beta sheet-rich aggregates. Conclusion. Secondary structure elements such as alpha-helices, beta sheets, turns, and loops are fundamental to protein architecture. Their local folding patterns influence the overall structure, stability, and function of proteins. Recognizing these patterns is essential for understanding protein folding, function, and the implications of disease-related mutations. References. 1. Eisenhaber, Frank, Prediction of Protein Function, 2 Basic Concepts and 1 Practical Recipe. Landis Bioscience and Springer Science Business Media, 2006. 2. Additional sources. Section 4. Protein tertiary structure, 3-dimensional folding. Introduction to protein tertiary structure. The tertiary structure of a protein is its overall 3-dimensional shape, formed by the folding of its polypeptide chain. This structure is essential as it determines the protein's functionality, specificity, and interactions. The tertiary structure is stabilized by various interactions between amino acid side chains, including hydrophobic interactions, hydrogen bonds, ionic bonds, and disulfide bridges. Stabilizing interactions in tertiary structure. 1. Hydrophobic interactions. Non-polar amino acid side chains cluster together in the interior of the protein, avoiding the aqueous environment. Hydrophobic collapse. This clustering is often an initial step in protein folding. 2. Hydrogen bonds, form between polar side chains and between backbone atoms. Helps stabilize secondary structure elements, alpha-helices and beta sheets, and tertiary folds. 3. Ionic bonds, salt bridges, occur between positively charged, e.g. lysine, arginine, and negatively charged, e.g. aspartate, glutamate side chains. Play a significant role in stabilizing the protein surface. 4. Disulfide bridges. Covalent bonds between cysteine residues form disulfide bridges, stabilizing the protein's tertiary structure. Common in extracellular proteins exposed to oxidative environments. 5. Van der Waals forces. Weak interactions between nonpolar side chains in close proximity contribute to overall stability and packing density. Domains, functional and structural units. Proteins often contain distinct structural and functional units called domains. A domain is an independently folding unit, often associated with a particular function. 1. Types of domains. Functional domains directly involved in specific biochemical functions, e.g. DNA binding, enzymatic activity. Structural domains provide structural stability and scaffolding. Topological domains, regions separated by membrane barriers or subcellular compartments. 2. Common domain families. Immunoglobulin Ig domains, found in antibodies and other immune proteins. Kinase domains phosphorylate substrates in signal transduction pathways. SH2 and SH3 domains involved in protein-protein interactions. Zinc finger domains. mediate DNA binding through coordination with zinc ions. 3. Domain databases. FAM, collection of protein families and domains. SMART, specialized domains in signaling, nuclear, and extracellular proteins. ProSite, motif-based domain information. Folding pathways, chaperones, and hydrophobic collapse. The folding of proteins into their native tertiary structure is a highly regulated process involving several key steps and often the assistance of molecular chaperones. 1. Hydrophobic collapse, the initial step in protein folding where hydrophobic side chains aggregate, forming a molten globule. Secondary structure elements form during this stage. 2. Folding pathways and intermediate states. Proteins may fold through different pathways, often involving intermediate states. Molten globule, a partially folded state with native-like secondary structure but disordered tertiary structure. 3. Molecular chaperones, assist in proper protein folding and prevent aggregation. Heat shock proteins, HSPs, stabilize partially folded intermediates. Chaperonins, large barrel-shaped complexes, GRO-ES, that provide a folding environment. Protein disulfide isomerase, PDI, facilitates the formation and rearrangement of disulfide bonds. Peptidyl prolyl isomerase, PPI, accelerates the cis-trans isomerization of proline residues. 4. Folding challenges. Aggregation. Misfolded proteins can form insoluble aggregates. Protein misfolding diseases, Alzheimer's, Parkinson's, and Huntington's diseases, are linked to protein misfolding and aggregation. Post-translational modifications. Post-translational modifications, PTMs, are crucial for regulating protein structure and function. They often occur after the protein has folded into its tertiary structure. 1. Phosphorylation, addition of phosphate groups to serine, threonine, or tyrosine residues, regulates enzyme activity, protein-protein interactions, and signal transduction. 2. Glycosylation, attachment of carbohydrate chains to asparagine, N-linked, or serine-threonine, O-linked residues, essential for protein stability, folding, and cell-cell recognition. 3. Lipidation, covalent attachment of lipid groups, anchoring proteins to membranes. Myristoylation, addition of a myristoyl group to the N-terminal glycine. Palmitoylation, attachment of palmitic acid to cysteine residues. Prenylation, addition of farnesyl or geranyl groups. 4. Ubiquitination, covalent attachment of ubiquitin to lysine residues, marking proteins for degradation. Involved in protein turnover via the ubiquitin proteasome system. 5. Acetylation, addition of acetyl groups to lysine residues, affecting protein stability and interactions. 6. Methylation, methylation of lysine and arginine residues, regulating gene expression and protein-protein interactions. 7. Disulfide bond formation, stabilizes the tertiary structure of extracellular proteins. Significance of tertiary structure in protein function. 1. Enzymatic activity. Proper tertiary structure is crucial for the formation of enzyme active sites. Example, serine proteases, e.g. trypsin, have active sites formed by the catalytic triad. 2. Molecular recognition. Protein-ligand and protein-protein interactions rely on specific tertiary structures. Example, hemoglobin undergoes conformational changes upon oxygen binding. 3. Signal transduction. Many signaling proteins undergo Section 4. Protein tertiary structure, three-dimensional folding. Introduction to protein tertiary structure. The tertiary structure of a protein is its overall three-dimensional shape, formed by the folding of its polypeptide chain. This structure is essential as it determines the protein's functionality, specificity, and interactions. The tertiary structure is stabilized by various interactions between amino acid side chains, including hydrophobic interactions, hydrogen bonds, ionic bonds, and disulfide bridges. Stabilizing interactions in tertiary structure. Section 5. Protein Quaternary structure, multimeric complexes. Introduction to protein quaternary structure. While many proteins function as individual polypeptides, a significant proportion require multiple subunits to assemble and operate effectively. The quaternary structure of proteins refers to the arrangement and interaction of these subunits, creating multimeric complexes. The architecture and dynamic nature of these complexes are crucial for the regulation of their biological functions. Types of multimeric proteins. 1. Homodimers and heterodimers. Homodimers, complexes formed by two identical subunits. Example, lactate dehydrogenase, a homodimeric enzyme involved in glycolysis. Heterodimers, complexes formed by two different subunits. Example, hemoglobin, a heterotetramer with two alpha and two beta subunits. 2. Larger homomeric complexes, homotetramers, four identical subunits. Example, catalase, an enzyme that catalyzes the decomposition of hydrogen peroxide. Homohexamers, six identical subunits. Example, glutamine synthetase, an enzyme involved in nitrogen metabolism. Homoheptamers and larger oligomers. Example, proteasomes, large complexes that degrade ubiquitinated proteins. 3. Larger heteromeric complexes, heterooligomers, complexes with varying subunits. Example, ATP synthase, a membrane-bound enzyme complex responsible for ATP production. Complex multiprotein assemblies. Example, ribosomes, which consist of rRNA and proteins forming a functional translation machinery. Cooperative binding and allosteric regulation. In multimeric proteins, the binding of a ligand to one subunit can influence the binding affinity of neighboring subunits, a phenomenon known as cooperativity. Allosteric regulation involves conformational changes that alter protein activity. 1. Cooperative binding. Positive cooperativity. Binding of the first ligand increases the affinity for subsequent ligands. Example, hemoglobin exhibits positive cooperativity for oxygen binding. Negative cooperativity. Binding of the first ligand decreases the affinity for subsequent ligands. Example, glucokinase, a key enzyme in glucose metabolism. Allosteric regulation. Allosteric sites. Binding of effectors at sites distinct from the active site induces conformational changes. T and R states, tense state, low affinity conformation. Relaxed R state, high affinity conformation. Examples, hemoglobin. Oxygen binding shifts hemoglobin from the T state to the R state. Aspartate transcarbamylase, ACASE, catalyzes the first step of pyrimidine biosynthesis and is regulated allosterically by ATP and CTP. 3 models of allosteric regulation. Monod-Wyman-Shanju, MWC model. All subunits transition simultaneously between T and R states. Koshland-Nemethy-Filmer, KNF model. Subunits transition sequentially, inducing conformational changes. Protein-protein interactions and modular domains. Proteins often recognize and bind to specific sequences or structural motifs in their partners. Such interactions are mediated by modular domains, allowing the formation of protein complexes. 1. S.H.2, Sersi homology 2 domain, recognizes phosphorylated tyrosine residues within specific motifs, found in signaling proteins such as kinases and phosphatases. Example, the S.H.2 domain of the Src kinase binds phosphotyrosine and receptor tyrosine kinases. 2. S.H.3, Ars homology 3 domain, recognizes proline-rich motifs, PXXP, involved in signaling pathways, cytoskeletal organization, and membrane trafficking. Example, the SH3 domain of the GRB2 adapter protein binds proline-rich regions in SOS, a RAS activator. 3-PH, plextrin homology domain, binds phosphoinositides and proteins involved in signaling pathways. Example, the pH domain of ACT binds PIP3, localizing it to the plasma membrane for activation. 4. PDZ domain, postsynaptic density 95, discs large, zonula occlusions 1, recognizes C-terminal sequences of target proteins. Example, the PDZ domain of PSD95 binds the C-terminus of NMDA receptors in the synapse. 5. Leucine zipper, involved in DNA binding and dimerization of transcription factors. Example, the leucine zipper motif in CFOS and C-JUN forms a heterodimeric transcription factor, AP1. 6. EF hand motif, a helix-loop-helix motif that binds calcium ions. Example, calmodulin contains 4 EF hand motifs that enable calcium-dependent regulation. Assembly and stability of protein complexes. 1. Subunit interactions. Hydrophobic interactions. Major driving force for subunit association. Hydrogen bonds and salt bridges contribute to specificity and stability. Disulfide bridges. Covalent bonds between subunits, e.g. immunoglobulins. 2. Molecular chaperones. Assist in the proper assembly of multimeric complexes. Example, HSP90 stabilizes steroid hormone receptors before binding ligands. 3. Post-translational modifications. Modifications such as phosphorylation, glycosylation, and ubiquitination can regulate complex assembly and activity. Functional significance of quaternary structure. 1. Enzymatic regulation. Enzymes with quaternary structures often exhibit allosteric regulation. Example, phosphofructokinase PFK, a key enzyme in glycolysis. 2. Signal transduction. Signal transduction pathways rely on the formation of multi-protein complexes. Example, MAP kinase signaling involves the sequential phosphorylation of a kinase cascade. 3. Molecular recognition. Protein complexes form specific recognition interfaces. Example, antigen-antibody binding relies on quaternary structure interactions. 4. Structural support. Multimeric proteins contribute to Section 5. Protein quaternary structure, multimeric complexes. Introduction to protein quaternary structure. While many proteins function as individual. Section 6. Protein folding and stability. Introduction to protein folding and stability. Proteins are linear polymers of amino acids that must fold into specific three-dimensional structures to function properly. Protein folding is a complex and highly regulated process involving molecular chaperones, folding intermediates, and stabilization factors. Misfolding can lead to aggregation and diseases like Alzheimer's. Here, we delve into the intricacies of protein folding, the role of chaperones, and factors influencing stability. Molecular chaperones, heat shock proteins, and chaperonins. Molecular chaperones are essential for protein folding and maintenance of protein homeostasis, proteostasis. They assist in folding newly synthesized polypeptides, refolding misfolded proteins, and preventing aggregation. 1. Heat shock proteins, HSPs. Heat shock proteins are a group of chaperones induced under stress conditions, such as heat or oxidative stress. They can be grouped into different families based on their molecular weight. HSP70 family. Bind to nascent polypeptides and misfolded proteins, preventing aggregation. Mechanism. ATP binding and hydrolysis regulate substrate binding and release. Co-chaperones, HSP40 stimulates HSP70 ATPase activity, while nucleotide exchange factors, EFs, facilitate ADP release. Example, DNAK, bacterial, BIAP, ER-specific, HSC70, cytosolic. HSP90 family play a critical role in stabilizing steroid hormone receptors and kinases. Mechanism. ATP, AT binding, and hydrolysis lead to conformational changes. HSP90 forms a molecular clamp around client proteins. Co-chaperones. P23 stabilizes the ATP-bound state, while CDC37 assists in kinase folding. Example, HTPG bacterial, GRP94, ER-specific, TRAP1 mitochondrial. Small heat shock proteins, HSPs, form oligomeric complexes that bind and stabilize unfolded proteins, function as holdases, preventing aggregation until refolding or degradation. Example, alpha-crystalline, lens protein, HSP27. 2. Chaperonins. Chaperonins are large, cylindrical complexes providing an isolated environment for protein folding. Group 1 chaperonins, found in prokaryotes, mitochondria, and chloroplasts. Composed of GRO-EL, double ring structure, and GRO-ES, lid-like co-chaperone. Mechanism. ATP binding leads to conformational changes in GRO-EL, allowing substrate binding. GRO-ES binding encloses the substrate, facilitating folding. ATP hydrolysis releases GRO-ES and the folded protein. Example, GRO-EL, GRO-ES in bacteria, HSP60, HSP10 in mitochondria. Group 2 chaperonins, found in archaea and eukaryotic cytosol. Subunits have a built-in lid domain that closes the cavity upon ATP binding. Example, TRIT-CCT in eukaryotes. 3. Protein disulfide isomerase, PDI, catalyzes the formation and isomerization of disulfide bonds in the endoplasmic reticulum, essential for proper folding of secretory and membrane proteins. 4. Peptidyl prolyl isomerase, PPI, accelerates the cis-trans isomerization of proline residues, important for the folding of proteins with proline-rich regions. Protein aggregation and misfolding diseases. Protein aggregation occurs when misfolded proteins aggregate into insoluble complexes. Aggregation is implicated in various neurodegenerative diseases. 1. Prions and amyloid diseases. Prions, infectious proteins that cause transmissible spongiform encephalopathies, TSEs. Example, Creutzfeldt-Jakob disease, CJD, Kuru, mad cow disease. Mechanism. Prion protein PRP misfolds from the normal PRP-C alpha helical to the infectious PRP-SC beta sheet-rich form. PRP-SC aggregates into amyloid fibrils and induces the conversion of PRP-C to PRP-SC. Amyloid diseases. Characterized by the accumulation of amyloid fibrils composed of beta sheet-rich proteins. Example, Alzheimer's disease, Parkinson's disease, Huntington's disease. Alzheimer's disease. Accumulation of amyloid beta, A-beta plaques, and tau protein neurofibrillary tangles. A-beta is derived from amyloid precursor protein, APP, by beta and gamma secretases. Parkinson's disease. Accumulation of alpha-synuclein into Lewy bodies. Huntington's disease. Expansion of CAG repeats in the Huntington gene leads to polyglutamine tract aggregation. 2. Protein folding diseases, cystic fibrosis, deletion of phenylalanine at position 508, delta 5008, in the CFTR protein, leads to misfolding and degradation of CFTR, affecting chloride ion transport. Tay-Sachs disease, mutation in the hexa gene affects the beta-hexosaminidase A enzyme. Accumulation of GM2 ganglioside causes neurodegeneration. 3. Protein quality control and degradation. Ubiquitin proteasome system. Ubiquitin tags misfolded proteins for degradation by the proteasome. E1, E2, E3 enzyme cascade. E1, activating enzyme. E2, conjugating enzyme. E3, ligase. Autophagy, degradation of damaged organelles and protein aggregates via lysosomes. Macroautophagy, encloses substrates in autophagosomes. Chaperone-mediated autophagy, HSP70 targets proteins to lysosomes. Stabilization factors, hydrophobic effect, disulfide bonds, and metal ions. Proteins rely on various stabilization factors to maintain their structure and resist unfolding or aggregation. 1. Hydrophobic effect. Nonpolar amino acids aggregate to avoid the aqueous environment, driving protein folding. Forms a hydrophobic core that stabilizes the protein's tertiary structure. Section 6. Protein folding and stability. Introduction to protein folding and stability. Proteins are linear polymers. Section 7. Protein function, enzymatic activity. Introduction to enzymatic activity. Enzymes are biological catalysts that accelerate biochemical reactions by lowering the activation energy required. They are essential for metabolic processes, DNA replication, signal transduction, and many other biological functions. Each enzyme has a specific structure, particularly at its active site, that determines its catalytic mechanism and specificity. Active sites, structure, and catalytic mechanisms. The active site is the region of an enzyme where substrate binding and catalysis occur. Its specific shape and chemical environment enable the enzyme to recognize and convert particular substrates into products. 1. Structure of active sites. Binding pocket, formed by amino acid residues that interact with the substrate. Catalytic residues, amino acids directly involved in the catalytic reaction. 2. Catalytic mechanisms, acid-base catalysis. Proton donors and acceptors, e.g. histidine, facilitate proton transfer. Example, RNase A uses histidine residues as acid and base catalysts. Covalent catalysis. A transient covalent bond forms between the enzyme and the substrate. Example, serine proteases, e.g. chymotrypsin, use a serine residue to form an acyl enzyme intermediate. Metal ion catalysis. Metal ions stabilize charged intermediates or facilitate redox reactions. Example, carbonic anhydrase uses a zinc ion to facilitate the conversion of CO2 to bicarbonate. Proximity and orientation. Enzymes bring substrates into close proximity and correct orientation. Example, DNA polymerase aligns nucleotides for accurate DNA synthesis. 3. Serine protease mechanism. Catalytic triad. Serine, histidine, and aspartate residues form a catalytic triad. Mechanism. Nucleophilic attack. Serine's hydroxyl group attacks the peptide bond of the substrate, forming an acyl enzyme intermediate. Tetrahedral intermediate, transition state stabilized by an oxyanion hole. Hydrolysis, water attacks the acyl enzyme, releasing the product. Enzyme kinetics, Michaelis-Menten kinetics, Km, and Vmax enzyme kinetics describes the rate at which enzymes catalyze reactions and the factors influencing these rates. The Michaelis-Menten model is widely used to characterize enzyme kinetics. 1. Michaelis-Menten kinetics. Model assumptions. Enzyme E binds to substrate S to form an enzyme-substrate complex ES. The ES complex can either dissociate back to E and S or proceed to form product P. The rate of product formation, VSER, depends on the concentration of ES. Reaction scheme, E plus S, right-left harpoons ES, right arrow, E rate equations. V0 equals a frac, V max S, KME, initial reaction velocity, Vmax, maximum velocity achieved at saturating substrate concentration, Km, Michaelis constant, the substrate concentration at which the reaction rate is half of Vmax. 2. Significance of Km and Vmax. Km represents the enzyme's affinity for its substrate. Lower Km indicates higher affinity. Vmax reflects the turnover number, kcat, the number of substrate molecules converted to product per enzyme molecule per second. Vmax equals kcat, total. Catalytic efficiency, K, measures an enzyme's specificity and efficiency. Higher kcat column indicates better catalytic performance. 3. Lineweaver-Burke plot. A double reciprocal plot used to determine Km and Vmax. Frac 1 V0 equals frac Km Vmax S plus frac 1 Vmax. The slope is Km per Vmax, and the y-intercept is 1 over Vmax. 4. Non-Michaelis-Menten kinetics. Allosteric enzymes do not follow Michaelis-Menten kinetics, exhibit sigmoidal S-shaped kinetics due to cooperative substrate binding. Inhibition and regulation. Enzymes are regulated by inhibitors and allosteric modulators to fine-tune their activity in response to cellular needs. 1. Types of inhibitors. Competitive inhibition. Inhibitor resembles the substrate and competes for binding at the active site. Effect on kinetics. Increases Km, decreases affinity. Vmax remains the same. Example, methotrexate inhibits dihydrofolate reductase by mimicking folate. Non-competitive inhibition. Inhibitor binds to an allosteric site, not the active site. Effect on kinetics. Decreases Vmax. Km remains the same. Example, allosteric inhibitors of fructose 1,6-bisphosphatase. Uncompetitive inhibition. Inhibitor binds only to the enzyme substrate complex, preventing product formation. Effect on kinetics. Decreases both Vmax and Km. Example, inhibition of placental alkaline phosphatase by phenylalanine. Mixed inhibition. Inhibitor can bind to both the enzyme and the enzyme substrate complex. Effect on kinetics. Decreases Vmax. Effect on Km varies, increase or decrease. 2. Allosteric regulation. Allosteric enzymes have multiple subunits and regulatory sites. Allosteric modulators, activators, increase enzyme activity. Inhibitors decrease enzyme activity. Sigmoidal kinetics. Cooperative substrate binding leads to a sigmoidal velocity versus substrate concentration curve. Example, phosphofructokinase 1, PFK1, regulated by ATP and AMP. 3. Feedback inhibition. The end product of a metabolic pathway inhibits an earlier step. Example, ATP inhibits phosphofructokinase in glycolysis. 4. Covalent modification. Section 7. Protein function, enzymatic activity. Introduction to enzymatic activity. Enzymes are biological catalysts that accelerate biochemical reactions by lowering the activation energy required. They are essential for metabolic processes, DNA replication, signal transduction, and many other biological functions. Each enzyme has a specific structure, particularly at its active site. Section 8. Protein function, structural proteins. Introduction to structural proteins. Structural proteins provide essential support and shape to cells, tissues, and organs. They form intricate networks within the cell and contribute to the extracellular matrix, ensuring the integrity and organization of tissues. Here, we delve into the different classes of structural proteins, focusing on cytoskeletal proteins, extracellular matrix proteins, and adhesion proteins. Cytoskeletal proteins, actin, tubulin, and intermediate filaments. The cytoskeleton is a dynamic network of protein filaments that provides structural support, enables cellular movement, and organizes intracellular components. It comprises 3 main types of filaments: actin filaments, microtubules, and intermediate filament. 1. Actin filaments, microfilaments. Structure and composition. Actin filaments are composed of globular actin, G-actin, subunits that polymerize into filamentous actin, F-actin. F-actin has polarity with a fast-growing plus end and a slow-growing nice end. Functions. Cell shape and structure. Form the cortical actin network beneath the plasma membrane. Cell movement. Involved in cell motility via lamellipodia and filopodia. Muscle contraction. Interact with myosin to enable muscle contraction. Intracellular transport. Serve as tracks for myosin motor proteins. Regulation of actin dynamics. Nucleating proteins. Formins and ARP23 complex nucleate new actin filaments. Severing proteins. Cofilin and gelsolin sever actin filaments, increasing turnover. Capping proteins. Cap Z and gelsolin cap filament ends, preventing further polymerization. Bundling proteins. Fimbrin and alpha-actinin cross-link actin filaments into bundles. Associated proteins. Myosin. Actin motor protein responsible for muscle contraction and intracellular transport. Tropomyosin and troponin regulate actin-myosin interactions in muscle. Microtubules. Structure and composition. Microtubules are hollow tubes composed of alpha and beta tubulin dimers. Exhibit polarity with a fast-growing plus end and a slow-growing minus end. Functions. Cell shape and support. Provide structural support and maintain cell shape. Intracellular transport. Serve as tracks for motor proteins kinesin and dynein. Cell division. Form the mitotic spindle during cell division. Cilia and flagella, core components of these modal structures. Regulation of microtubule dynamics, microtubule-associated proteins, stabilize microtubules and promote assembly, e.g. tau MAP2. Severing proteins. Catenin severs microtubules, regulating length and turnover. Capping proteins. Gamma-tubulin ring complex, gamma-TURC, nucleates and caps microtubules at the centrosome. Motor proteins. Kinesin moves cargo towards the plus end of microtubules, anterograde transport. Dynein moves cargo towards the minus end, retrograde transport. 3. Intermediate filaments. Intermediate filaments provide mechanical strength to cells and tissues, forming a stable and flexible network. Structure and composition. Composed of various proteins depending on the cell type, e.g. keratins, vimentin, neurofilaments. Lack polarity and do not have associated motor proteins. Functions. Mechanical support. Provide tensile strength to cells, protecting them from mechanical stress. Nuclear integrity. Form the nuclear lamina, lamins, providing structural support to the nuclear envelope. Types of intermediate filaments. Keratins, found in epithelial cells, providing protection against mechanical and chemical damage. Vimentin, present in mesenchymal cells, supporting cellular integrity. Neurofilaments, maintain the shape and caliber of axons in neurons. Lamins, form the nuclear lamina, regulating nuclear structure and gene expression. Extracellular matrix proteins, collagen, elastin, and fibronectin. The extracellular matrix, ECM, is a complex network of proteins and polysaccharides that provides structural support to tissues, facilitates cell signaling, and regulates cell behavior. 1. Collagen, structure and composition. Collagens are the most abundant proteins in the ECM, forming triple helical structures, composed of 3 polypeptide chains, alpha chains, forming a right-handed triple helix. Glycine is present at every third position, enabling tight packing of the helices. Types and distribution. Fibrillar collagens, type 1, 2, 3, form fibrils that provide tensile strength to tissues. Non-fibrillar collagens, type 4, SIC, form networks or associate with fibrillar collagens. Functions. Structural support. Provide tensile strength to skin, bone, cartilage, and tendons. Cell adhesion. Bind to integrins and other adhesion proteins, mediating cell-ECM interactions. Biosynthesis and assembly. Collagen precursors are synthesized as procollagen in the endoplasmic reticulum. Procollagen modification. Hydroxylation of prolinolene residues requires vitamin C. Glycosylation of hydroxylysine residues. Formation of intrachain and interchain disulfide bonds. Extracellular processing. Cleavage of N and C-terminal propeptides by procollagen peptidases. Fibril formation via lateral association and cross-linking. Collagen disorders. Osteogenesis imperfecta. Mutations in type 1 collagen genes lead to brittle bones. Ehlers-Danlos syndrome. Mutations in collagen or modifying enzymes result in joint hypermobility and skin fragility. 2. Elastin. Structure and composition. Elastin is a highly elastic protein found in connective tissues that require flexibility, composed of hydrophobic domain. Section 8. Protein. Section 9. Protein function, transport and signaling. Introduction to transport and signaling proteins. Transport proteins and signaling molecules are integral to maintaining cellular homeostasis and coordinating complex biological processes. They ensure the efficient movement of ions, molecules, and information across membranes and within cells. Here we explore different classes of transport proteins and signaling molecules, focusing on their mechanisms and roles. Transport proteins, ion channels, transporters, and pumps. Transport proteins facilitate the movement of ions, small molecules, and macromolecules across cell membranes. 1. Ion channels. Ion channels are integral membrane proteins that form pores through which ions passively diffuse down their electrochemical gradients. Structure and composition. Composed of multiple subunits forming a central pore. Most channels are highly selective for specific ions. Types of ion channels. Voltage-gated channels, open or close in response to changes in membrane potential. Example, voltage-gated sodium channels initiate action potentials in neurons. Ligand-gated channels, open in response to the binding of a ligand, neurotransmitter, or ion. Example, nicotinic acetylcholine receptor, NACHR, opens in response to acetylcholine binding. Mechanosensitive channels, respond to mechanical stimuli such as stretch or pressure. Example, piezo-1 channels are involved in touch and hearing. Aquaporins, facilitate the transport of water across cell membranes. Example, aquaporin 2 in the kidney is regulated by antidiuretic hormone. Ion channel disorders. Cystic fibrosis. Mutation in the CFTR chloride channel impairs ion transport in epithelial cells. Long QT syndrome. Mutations in voltage-gated potassium channels lead to cardiac arrhythmias. 2. Transporters, carriers. Transporters undergo conformational changes to move substrates across membranes, often against their concentration gradients. Types of transporters. Facilitated transporters, uniporters, move substrates down their concentration gradient without ATP. Example, GLUT1 transports glucose into erythrocytes. Co-transporters, symporters, and antiporters. Symporters move two substrates in the same direction. Antiporters move two substrates in opposite directions. Example, sodium-glucose cotransporter, SGLT1, reabsorbs glucose in the kidney. Active transporters, pumps, use ATP to move substrates against their concentration gradient. Major classes of active transporters. P-type ATPases undergo phosphorylation during transport. Example, N-phage Kpase ATPase maintains ion gradients by pumping N-apase out and Kpase into the cell. V-type ATPases pump protons into organelles, acidifying them. Example, V-ATPase acidifies lysosomes for degradation. ABC transporters. ATP-binding cassette transporters have two ATP-binding domains and two transmembrane domains. Example, P-glycoprotein MDR1 exports hydrophobic drugs from cancer cells. Transporter disorders, familial hypercholesterolemia, mutations in the LDL receptor impair cholesterol uptake. Hartnup disease, defective neutral amino acid transporter leads to amino acid malabsorption. 3. Pumps, primary active transporters. Pumps use ATP to transport ions and molecules across membranes. Na-Kmi ATPase maintains the sodium and potassium gradients across the plasma membrane. Pumps 3 Na ions out of the cell and 2 Kos ions into the cell per ATP. Crucial for generating the resting membrane potential and secondary active transport. Ca-SPY ATPases maintain low cytosolic calcium levels by pumping Ca2 ions into the sarcoplasmic reticulum, extracellular space Ca. HKATA-Ase secretes protons into the stomach, maintaining acidic gastric pH. Target of proton pump inhibitors used to treat acid reflux. Signaling molecules, hormones, cytokines, and growth factors. Signaling molecules convey information between cells, coordinating growth, differentiation, metabolism, and immune responses. 1. Hormones. Hormones are chemical messengers secreted by endocrine glands and act on distant target organs. Types of hormones. Peptide hormones, composed of amino acids and bind to surface receptors. Example, insulin regulates glucose uptake and metabolism. Steroid hormones, derived from cholesterol and bind to intracellular receptors. Example, cortisol mediates stress responses via glucocorticoid receptors. Amino acid-derived hormones, derived from tyrosine or tryptophan. Example, thyroid hormones T3, T4 regulate metabolism. Hormone disorders, diabetes mellitus. Insulin deficiency or resistance leads to hyperglycemia. Cushing's syndrome. Excess cortisol production results in weight gain and hypertension. 2. Cytokines. Cytokines are small proteins that regulate immune and inflammatory responses. Types of cytokine. Interleukins regulate immune cell differentiation and activity, e.g. IL-2, IL-6. Interferons induce antiviral states in cells, e.g. IFFN, IFN guanine. Tumor necrosis factor, TNF, mediates inflammation and apoptosis. Chemokines direct immune cell migration, e.g. CXCL8. Cytokine disorders, autoimmune diseases. Excess cytokine. Section 9. Protein function, transport and signaling. Introduction to transport and signaling proteins. Transport proteins and signaling molecules are integral to maintaining cellular homeostasis and coordinating complex biological processes. They ensure the efficient movement of ions, molecules, and information across membranes and within cells. Here we explore different classes of transport proteins and signaling molecules, focusing on their mechanisms and roles. Transport proteins, ion channels, transporters, and pumps. Transport proteins facilitate the movement of ions, small molecules, and macromolecules across cell membranes. 1. Ion channels. Ion channels are integral membrane proteins that form pores through which ions passively diffuse down their electrochemical gradients. Structure and composition. Composed of multiple subunits, forming a central pore. Section 10. Protein function, regulation, and degradation. Introduction to protein regulation and degradation. Proteins are essential to virtually every cellular process, and their functions must be tightly regulated to ensure proper cellular activity. Protein regulation involves post-translational modifications that modulate function, stability, and localization. Degradation mechanisms, such as the ubiquitin proteasome system and autophagy, are crucial for quality control and protein turnover. Here we discuss the different forms of protein regulation and degradation pathways in detail. Protein regulation, phosphorylation, ubiquitination, acetylation. Post-translational modifications, PTMs, regulate protein activity, stability, and interactions, allowing rapid cellular response to environmental cues. 1. Phosphorylation. Phosphorylation is the reversible addition of a phosphate group to serine, threonine, or tyrosine residues. Enzymes involved, kinases, transfer phosphate groups from ATP to specific amino acids. Phosphatases, remove phosphate groups, reversing phosphorylation. Functional effects. Enzyme activation inhibition alter catalytic activity glycogen phosphorylase. Protein-protein interactions. Phosphorylated residues can recruit SH2 or PTB domain-containing proteins. Subcellular localization. Regulates nuclear cytoplasmic transport, e.g. FOXO transcription factors. Major phosphorylation pathways. MAP kinase pathway, ERK, JNK, and P38 kinases phosphorylate transcription factors in response to growth factors and stress. PI3K ACT pathway, ACT phosphorylates proteins involved in cell survival and metabolism. mTOR pathway, mTOR phosphorylates proteins regulating translation and autophagy. Phosphorylation disorders. Cancer, overactive kinases, e.g. BCR-ABL, EGFR, drive uncontrolled cell growth. Diabetes, impaired insulin receptor signaling due to defective kinase activity. 2. Ubiquitination. Ubiquitination involves the attachment of ubiquitin, a small regulatory protein, to lysine residues of target proteins. Enzymes involved. E1, ubiquitin activating enzyme, activates ubiquitin via ATP-dependent adenylation. E2, ubiquitin conjugating enzyme, transfers ubiquitin from E1 to E3. E3, ubiquitin ligase, facilitates the transfer of ubiquitin to the target protein. Types of ubiquitination. Monoubiquitination, single ubiquitin attached to a protein, often regulating localization. Polyubiquitination, chains of ubiquitin linked via lys48 or lys63. Lys48 linked chains, target proteins for proteasomal degradation. Lys63 linked chains, involved in DNA repair and signaling. Functional effects. Proteasomal degradation, lys48 linked chains target proteins for degradation. Endocytosis and trafficking. Monoubiquitination regulates receptor endocytosis. DNA repair and signaling. Lys63 linked chains recruit DNA repair proteins. Ubiquitination disorders. Parkinson's disease, mutations in Parkin E3 ligase impair mitophagy. Angelman syndrome. Mutation in UBE3A, E3 ligase leads to neurodevelopmental defects. Acetylation. Acetylation involves the addition of an acetyl group to lysine residues, affecting protein stability and interactions. Enzymes involved. Histone acetyltransferases, HATs, acetylate histones and non-histone proteins. Histone deacetylases, HDACs, remove acetyl groups, reversing acetylation. Functional effects. Gene expression. Acetylation of histones relaxes chromatin structure, promoting transcription. Protein stability. Acetylation can enhance or reduce protein stability. Protein-protein interactions. Acetylated residues can recruit bromodomain-containing proteins. Acetylation disorders, cancer. Dysregulated acetylation due to mutations in HATS HDAX leads to aberrant gene expression. Huntington's disease. Reduced acetylation impairs histone function and gene expression. Proteasome and ubiquitin system, targeted degradation via ubiquitination. The ubiquitin proteasome system, UPS, is the primary pathway for targeted protein degradation, maintaining cellular proteostasis. 1. Ubiquitin proteasome system, UPS. Ubiquitination process, E1 activation. Ubiquitin is adenylated and transferred to E1 via a thioester bond. E2 conjugation. Ubiquitin is transferred from E1 to E2. E3 ligase interaction. E3 ligase binds to both the substrate and E2, facilitating ubiquitin transfer. Polyubiquitin chain formation. Multiple ubiquitin molecules are added to form a chain. Proteasome structure and function. 20s core particle, 4 stacked rings, 2 outer alpha rings and 2 inner beta rings. Beta subunits contain proteolytic sites with trypsin-like, chymotrypsin-like, and caspase-like activities. 19s regulatory particle recognizes ubiquitinated proteins and unfolds them for translocation into the 20s core. Contains ubiquitinases to remove ubiquitin chains before degradation. Proteasomal degradation process. Polyubiquitinated proteins are recognized and bound by the 19s regulatory particle. ATP-dependent unfolding and translocation into the 20s core occur. Proteins are degraded into small peptides by proteolytic subunits. 2. Proteasome inhibitors. Proteasome inhibitors are used in cancer therapy to induce apoptosis in tumor cells. Bortezomib reversibly inhibits the chymotrypsin-like activity of the proteasome. Carfilzomib irreversibly inhibits the chymotrypsin-like activity. 3. Dubiquinases, DUBs. DUBs remove ubiquitin chains, regulating substrate stability and recycling ubiquitin. USP family cysteine proteases that cleave ubiquitin chains, e.g. USP14, USP7. JAM family, zinc metalloproteases that disassemble ubiquitin chains, e.g. RPN11. Autophagy, degradation of damaged organelles and protein aggregates. Autophagy is a cellular degradation process that removes damaged organelles and protein aggregates, maintaining cellular health. 1. Types of autophagy. Macroautophagy. Double membrane vesicles, autophagosomes, enclose cellular material and fuse with lysosomes for degradation. Steps in macroautophagy. Nucleation, initiation complex, ULK1-AG13, recruits the PI3K complex, VPS34, BCLIN1. Elongation and closure. ATG proteins expand the phagophore membrane to form a double membrane autophagosome. Fusion with lysosome. Autophagosomes fuse with lysosomes to form autolysomes. Section 10. Protein function, regulation, and degradation. Conclusion. The central role of proteins. Proteins are indispensable to life, serving as the building blocks and workhorses of all living organisms. Their vast diversity in structure and function underlies every aspect of cellular processes. From structural support to catalytic activity, signaling, and transport, proteins orchestrate a multitude of biological functions. Proteins and their multifaceted roles. 1. Structural components. Structural proteins, such as actin, tubulin, and collagen, provide support and shape to cells and tissues. They form the cytoskeleton, anchor cells to the extracellular matrix, and enable cellular movements and muscle contraction. Without these proteins, cellular architecture would collapse. 2. Enzymatic catalysis. Enzymes are catalysts that accelerate biochemical reactions by lowering activation energy. They are vital for metabolic processes, DNA replication, and signal transduction. Understanding enzyme kinetics and regulation helps in the development of targeted therapeutics and diagnostic tools. 3. Transport and signaling. Transport proteins ensure the proper movement of ions and molecules across membranes. Signaling molecules, including hormones, cytokines, and growth factors, convey information between cells. Receptors and second messengers transmit these signals, coordinating cellular responses. 4. Regulation and degradation. Protein function is tightly regulated by post-translational modifications, such as phosphorylation, ubiquitination, and acetylation. Degradation pathways, including the ubiquitin proteasome system and autophagy, ensure protein quality control and turnover. The importance of protein structure. The structure of a protein determines its function. Each level of protein structure, from primary to quaternary, contributes to its unique shape and activity. 1. Primary structure. The linear sequence of amino acids dictates how the protein will fold and function. 2. Secondary structure. Local folding patterns, such as alpha-helices and beta sheets, form due to hydrogen bonding. 3. Tertiary structure. The overall three-dimensional shape is stabilized by hydrophobic interactions, hydrogen bonds, ionic bonds, and disulfide bridges. 4. Quaternary structure. Multimeric complexes with multiple subunits enhance cooperative binding and allosteric regulation. Understanding protein function and disease. Misfolding or mutations in proteins can lead to diseases. Examples include neurodegenerative diseases like Alzheimer's and Parkinson's, where protein aggregation disrupts cellular function. By understanding protein structure and function, scientists can identify disease mechanisms and develop targeted therapies. 1. Protein folding and misfolding. Molecular chaperones assist in proper folding, while the ubiquitin proteasome system and autophagy degrade misfolded proteins. Aggregation of misfolded proteins is linked to neurodegenerative diseases. 2. Genetic mutations and deficiency diseases. Mutations affecting enzyme function can lead to metabolic disorders, while mutations in structural proteins can cause connective tissue diseases. 3. Therapeutic targets. Proteins like kinases, GPCRs, and proteasomes serve as targets for drug development. Enzyme inhibitors and monoclonal antibodies are examples of successful protein-based therapeutics. Conclusion: unlocking insights into biology. Understanding the structure, function, and regulation of proteins unlocks insights into the fundamental workings of biology. Proteins are at the heart of cellular processes, from metabolism and signaling to immunity and development. By studying proteins, scientists can decipher disease mechanisms, identify biomarkers, and develop new therapies. As research advances, the central role of proteins in life continues to inspire new discoveries and innovations. References: 1. Eisenhaber, Frank. Prediction of Protein Function, 2 Basic Concepts and 1 Practical Recipe. Landis Bioscience and Springer Science Business Media, 2006. 2 additional sources.